Welcome to this session. In this session, we are going to learn about vesicular transport. Already we have seen the topics passive transport, active transport. So we know the main difference between passive and active transport is in passive transport, the lipid soluble substances from higher concentration to lower concentration without spending any energy, they'll move. That is passive transport. Where we have different types of passive transport mechanisms like diffusion, where even diffusion is again divided into simple diffusion, where the most of the lipid soluble substances like gases, we told oxygen, carbon dioxide, nitrogen, steroids, alcohol, and all those substances will be transported from higher concentration to the lower concentration. So that is simple diffusion. In case of even in simple diffusion, the water soluble substances as well as the lipid insoluble substances, they will transported from higher concentration to lower concentration when there is a presence of channel proteins, and these channel proteins are selectively permeable and they have gating mechanism. Based on that channel proteins are divided into voltage gated channels, mechanically gated channels, and the ligand gated channels. And when you come to the facilitated diffusion, it is also called as carrier mediated diffusion because it needs the carrier proteins. And this through this facilitated diffusion, most of the substances like glucose and amino acids, they'll be transported. And this carrier proteins are three different types: uniport, symport, and antiport. Only one substance is transported through uniport carrier protein, and in case of symport, two substances are transported in same direction, whereas in antiport, two substances are transported in opposite directions. So that is the facilitated diffusion, where we have seen main differences between simple and facilitated diffusion, and we came back to osmosis, which is again a type of passive transport. So what is osmosis? The movement of solvent from the lower concentration of solutes to the higher concentration of solutes, which can be prevented when we apply pressure on the area of higher concentration. So that is called as osmotic pressure, and the main particles which exerts osmotic pressure, which is also called as colloidal osmotic pressure, is the plasma proteins, especially the albumin, which is the main plasma protein, which exerts about 25 millimeters of mercury of oncotic pressure. So this is, we have seen up to passive transport, and yesterday we have seen active transport. And active transport is, it is the transport of substances against the gradient, that is from lower concentration to higher concentration, and with the expenditure of energy. Again, it is of two types: primary and secondary active transport. In primary active transport, is ATP is directly liberating the energy and it is utilized to transport substances. So that is active transport, and most of the active transport mechanisms are called as active pump mechanisms. And through active transport, ionic substances like sodium, chloride, hydrogen, calcium, and all these substances are transported, whereas the non-ionic substances like glucose, amino acids, even urea can be transported through active transport mechanisms. So we have seen primary active transport examples like sodium potassium pump, calcium ATPase pump, and then hydrogen potassium ATPase pump. So we have seen where these are present and what are the functions of these. The very important point to remember is about sodium potassium pump, which can be asked as a 5-mark question, and many MCQs can be asked from this sodium potassium pump. And we have seen it is a very important pump which helps to maintain the cell volume, and it is also an electrogenic pump. And the next thing is, we have seen about secondary active transport mechanisms, and in secondary active transport, it is linked with the sodium. So in transport of substances is linked with sodium in secondary active transport, which is again divided into sodium co-transport and sodium counter-transport. In case of sodium co-transport, we have seen some of the examples like sodium glucose co-transport and sodium amino acid transport. All these are usually seen in the renal tubule. And then sodium, the counter transport mechanisms like sodium calcium counter transport and sodium hydrogen counter transport, which is also seen in renal tubular cells. So up to this, we have seen the passive and active transport mechanisms, and today we are going to see one more transport mechanisms, and this is the end of this transport mechanisms across cell membrane. So vesicular transport, the name itself is telling that some vesicles are forming to transport substances. So vesicular transport. Vesicular transport mainly helps. There are some substances which cannot be passed through simple diffusion and even by active transport mechanism. So that type of substances are transported from through this vesicular transport mechanism, especially the transport of macromolecules such as large protein molecules, enzymes, neurotransmitters, the cholesterol, iron, all these substances are transported especially through this vesicular transport mechanism, and it is again the energy dependent process. It also requires ATP for its energy, and this vesicular transport is again divided into endocytosis and exocytosis. So endo, which means the substances are transported into the cell, whereas exocytosis, which means the substances are transported out of the cell. And in both of this, I told you the vesicles will be will releasing the substances. And endocytosis, again three types: pinocytosis, phagocytosis, and receptor mediated endocytosis. What is meant by pinocytosis? The intrusion of liquid particles, that is pinocytosis, which is also called as cell drinking. Whereas phagocytosis, we have seen when we studied lysosomes, the cells which are involved in phagocytic activity, for example like neutrophils and macrophages, they will have more number of lysosomes because they will destroy the foreign bodies like bacteria. They will ingest and the lysosomes will destroy them. Where the hydrolytic enzymes which are present in the lysosome, they will destroy them, and they'll be the destroyed particles will be thrown out of them through the exocytosis process. So here phagocytosis is an endocytic process where the substances like bacteria or the damaged tissues, all this will be engulfed by the engulfed by the cell membrane, and it will be pinched off from the cell membrane, and endocytic vesicle will be formed. And that endocytic vesicle, endophagolysosome, it will contain the lysosome particles, and these lysosomes will destroy the engulfed particles. So that is the process of phagocytosis, which we will study detailedly when we study about the neutrophils. So phagocytosis itself is a 5-mark question. So the third type is receptor mediated endocytosis, where in the cell membrane we will have some receptors which will help in the endocytosis process. So these three are the endocytosis, the types of endocytosis. So vesicular transport is of two types: endocytosis and exocytosis. Endocytosis is again three types: pinocytosis, phagocytosis, and receptor mediated endocytosis. So endocytosis, it is a process in which the substance is transported into the cell by infolding of the cell membrane around the substance and internalizing it. So pinocytosis, it is also called as cell drinking, where it is the process of engulfing of liquid substances by enfolding of the cell membrane. So the example for this pinocytosis is reabsorption by the renal tubular epithelial cells. What is phagocytosis? It is also called as cell eating, where it is the process of engulfment of solid particles such as bacteria, dead tissue, and foreign particles by the cells. So here you can see in the picture that pinocytosis, that is the intrusion of liquid particles. So the cell membrane forms cell membrane surrounds the particles, and it is internalizing it, and then it is the pinocytic vesicle is pinched off from the cell membrane. So the next process is phagocytosis, where you can see the cell membrane projects some pseudopodias, which are false feet, and then it will engulf the foreign bodies, and phagolysosomes are pinched up from the cell membrane, and this is how the phagocytosis will occur. So receptor mediated endocytosis, you can see there are some receptors which are present in the cell membrane. So the substance will be bind with the receptor. The substance receptor complex will be formed, which is lined by a special protein which is called as clathrin, and then this clathrin coated vesicle will be pinched off from the cell membrane. Once this clathrin is once this endocytic vesicle is formed, this clathrin will be fall off from the vesicle, and it will be reused for the next receptor mediated endocytosis process. So in this process, the substance is to be transported binds with the special receptor proteins present on the cell surface. The receptor protein substance complex is then engulfed by the cell membrane by the process of endocytosis. So the substances like iron and cholesterol, please remember this, substances like iron and cholesterol are transported by this receptor mediated endocytosis. So what is the mechanism of endocytosis? There are two pathways. One is constitutive pathway, the other one is clathrin mediated endocytosis or clathrin mediated endocytosis. So the constitutive pathway, where the molecule comes and makes contact with the cell membrane. So the non-cytoplasmic sites will be fused with the cell membrane, and the cell membrane invaginates and it pinches off and form the endocytic vesicle will be formed, and then it pinches off from the cell membrane. So these substances are transported into the cell. So this is how the constitutive pathway of endocytosis occurs. Whereas the clathrin mediated pathway, where the substance, the ligand will come and bind with the receptor which is present on the cell membrane, and there is a protein which is called as clathrin. This clathrin will form a N circle in the cell membrane. So this clathrin is a protein which is present in the cell membrane, where it will forms a N circle, and once the endocytic vesicle is formed, this clathrin will fall off from the endocytic vesicle, and it will be reused for the next cycle. So as the endocytosis progresses, clathrin molecule surrounds the endocytic vesicle. So here you can see the green color structure. So that is the substances which are called as clathrin proteins, and after complete formation of vesicle, the clathrin molecules falls off, recycled to form another vesicle. So what is exocytosis? Up to this we have seen endocytosis. The next thing is exocytosis. So substances are expelled from the cell without passing through the cell membrane. I told you exocytosis, which means the substances are transported out of the cell. So they will pass out of the cell without passing through the cell membrane. So the substances which are to be extruded collected in the form of granules or vesicles which moves towards the cell membrane. So they'll come, these secretory vesicle will come and fuse with the cell membrane, and the area of fusion breaks down, and it will release the contents like neurotransmitters, enzymes, proteins. All these will be released into the released out of the cell membrane by the process of this exocytosis, and it is a calcium dependent process. Please remember, the exocytosis requires calcium and energy to release the contents of the secretory vesicles out of the cell membrane, and the secretory vesicle will come and fuse with the cell membrane with the help of some proteins which are called as docking proteins. This we will study when we study about neuromuscular junction. There are some proteins which are called as V snare protein, T snare protein. There is a protein which is present in the membrane of the secretory vesicle which is called as T snare protein. So there is a protein which is called as synaptobrevin, which is a T snare protein which is a docking protein which binds or which links with the protein, the docking protein which is present in the axonal membrane, and this mechanism, the linking of these V snare and T snare protein like synaptobrevin and syntaxin, both of these will link, and the secretory vesicle will attach with the presynaptic membrane which is the axonal membrane, and this mechanism is called docking. Just remember this, we will study with the picture when we study about the neuromuscular junction. So these are the this is how the secretory vesicle will come and fuse with the cell membrane, and it will release its contents into the out of the cell membrane. So I told you there are enzymes and neurotransmitters, hormones, all these are secreted by the process of exocytosis, and excretion of specific hormones and granules by the cells is termed as amniocytosis. So here you can see the substances, secretory, within the secretory vesicles the substances are present. This secretory vesicle will approach and they will bind with they will attach with the specific areas of the cell membrane, and this is by the help of docking protein. So this mechanism is docking, and then this substances will be released out of the cell without there is some breaks will occur in the cell membrane which will release the substances out of the cell. So this is exocytosis. Answer for this MCQ questions: clathrin is used in exocytosis, receptor mediated endocytosis, cell to cell adhesion, in plasma membrane. So what is the answer? The answer is receptor mediated endocytosis. So clathrin is used in receptor mediated endocytosis. Pinocytosis is intrusion of solid particles, extrusion of liquid particles, intrusion of liquid particles, extrusion of solid particles. So what we should remember, pinocytosis is a process of endocytosis. So it must be intrusion, not extrusion. So we should remember, intrusion of solid particles is called as phagocytosis. So intrusion of liquid particles is called as pinocytosis. So the answer is C, which is intrusion of liquid particles. The exocytosis requires which ion? So exocytosis requires calcium. We will study detailedly about this exocytosis process, even detailedly when we study neuromuscular junction. Just remember, whenever there is increase in the intracellular calcium, what will happen? The secretory vesicle will go and bind with the cell membrane. The docking will occur, and then they will release by the process of this exocytosis, the secretory vesicle will release the contents of that secretory vesicles into the out of the cell membrane. Thank you.